So hi everyone, thanks for coming and then joining us to the Statistical and AI Methods for Health Data Science seminars by the Startup AI Alliance. So the goal of the seminar series is to introduce various kinds of biomedical data to the statistical community and then try to discuss current advances and then challenges in the fields of statistics and AI and the future directions. Just to give you a brief overview of the Startup AI group, so it aims to organize activities that empower statisticians to participate more actively in the AI research and then leadership. It's dedicated to enhancing the role of statisticians in addressing real-world challenges through AI research, and then emphasize the dual importance of leveraging both statistical methods and AI tools, ensuring that statisticians are not only participants, but also influential leaders in applying these combined approaches to solve critical problems. So right now, the Startup AI group has been officially recognized as an ASA interest group. So here's this website. So please feel free to sign up to the group, join the group, and then receive emails from the website. And then here's the steering committee of the group, and then also the inaugural officers. Today, it is my great honor and then pleasure to introduce Dr. Martin Lindquist to give the inaugural seminar. So Dr. Lindquist is a professor in the Department of Biostatistics at the Johns Hopkins School of Public Health. He is a pioneer researcher in statistical modeling of neuroimaging data and has been at the forefront of advancing methodological approaches in the field. He is also among the first to formally introduce causal inference and causal media analysis concepts to the neuroimaging research, which paved a lot of foundation to the causal research in this field. Beyond research, Dr. Lindquist also dedicated a lot of effort to education. His online course, Principles of fMRI, has been taken by over 100,000 students worldwide. So myself also took his online course when I was a student and then tried to step into this field. I think that would be a great intro to fMRI and neuroimaging study if you wanted to start working in this area. Dr. Lindquist's contribution has been widely recognized. He received the Education in Neuroimaging Award from the Organization of Human Brain Mapping, which sets a really great example of how a statistician can make a profound impact to the statistical community. To the science domain. So without further ado, so before I hand it over to Martin, so just a couple logistics. So please feel free to type questions in the chat box if you have a question. And then if you want to also ask questions, so please also raise your hand and then I will call your name and then ask the question. And then if you think that question is pretty, it's a long question and for discussion, so please save it to the end of the talk and then we will save some time for the discussion. So now without further ado, I will just hand it over to Martin and then Martin, it's all yours.
Oh, thanks, Yi. That was a generous introduction, and thank you all for attending. It's a really nice turnout here, and it's a really great pleasure to come here and speak to you. And I guess the first speaker in the seminar series, that's a great honor as well. So what I'm going to talk about is I'm going to talk a little bit about neuroimaging data, and with this particular focus on what's called functional neuroimaging data. I'm going to try to sort of walk through kind of what the data looks like and some of its characteristics that are important from a statistical point of view. I'm going to walk through some of the important questions that we seek to address with neuroimaging data. And since there's an AI focus to this, I want to then pivot to sort of looking at some examples of where AI has been utilized in neuroimaging and sort of kind of pitfalls and promises with AI. And then I also want to end up just talking about how you can get started if you're inspired by this kind of data and want to get your hands dirty. So I think that's the sort of the layout, the roadmap of the talk. But just to sort of get started, sort of understanding the brain is really one of the most important problems that are facing us today. Obama sort of famously called the century of the brain. We're going to finally understand how it works and how it functions and how it relates to different sort of disorders and the like. And so neuroimaging is kind of an umbrella term for kind of there's really an ever increasing number of kind of minimally invasive techniques for studying the brain. And these techniques can be used to measure brain structure, brain function, and sort of the progression of disease. And These techniques are being applied in a large number of sort of medical and scientific areas of inquiry. Lots of domains are sort of incorporating neuroimaging. So psychology incorporates a lot of neuroimaging, psychiatry, but even things like economics and marketing. So there's sort of a wide range of different disciplines that are sort of interested in utilizing neuroimaging and understanding how how the brain function sort of impacts their particular research. In general, I usually sort of separate neuroimaging into sort of two major categories, which I'm going to call structural and functional neuroimaging. And so there's a number of different ways of obtaining structural neuroimages and functional neuroimages. And so I'm going to walk you through that just to kind of give you, again, a little bit of a roadmap. So what is structural neuroimaging? Well, structural or neuroimaging is sort of akin to, the X-ray you get if you think that you broke a bone, right? This is a sort of a snapshot of your brain, and this is used to study, brain structure and also the diagnosis of disease or injury. So if you come into the ER and you were maybe in an accident, or where there might have been some head injury, or if you had, exhibiting sort of behaviors consistent with a stroke or a lesion, you might get a structural brain image, and then you can sort of look at it and see if there, what's going on, and if there's any abnormalities present. And so typical modalities are sort of the original one is sort of computed tomography or CT, that was previously known as CAT scans, and sometimes still referred as that. Magnetic resonance imaging or MRI, and positron emission tomography or PET. So these are different ways of sort of getting snapshots of the brain. And here's just a sort of cartoon image. Ideally we want to, you don't want to cut this guy up like this, but if you could get a photograph and you could see what his brain looks like. But we'd want to do this in a way that we don't have to actually do something that's invasive, which you could only do post-mortem. You would want to do it for a living subject. And so these are just three different modalities and these modalities, they sort of focus on different aspects of the brain. So some modalities are more sensitive to certain tissue types than others. Even something like MRI can be programmed in many ways so that it's sensitive to different. So these are a couple of different types of images. There's T1 weighted, T2 weighted, flare, proton density images. And as you can see here with these three different types, the contrast between different sort of tissue types, in particular sort of gray matter and white matter and CSF, is sort of highlighted in different ways depending on the contrast, right? So if you're interested in particular type of, if you're interested in segmenting the brain, there's particular sort of knobs that are more, if you're interested in finding sort of lesions, there's particular ways to measure the brain that might be more sensitive to that. Now, MRI is an extremely versatile technique. You can also study the diffusion of water molecules in the brain. And this is pretty cool. So at every location, you can sort of see what the most likely path of the water diffusion is. And you can use this to sort of create these white matter tracks. And these white matter tracks are a different type of structural image in that they kind of tell you where the roads are between different brain regions, right? This is telling you, what your brain looks like, and this is sort of telling you, is there a path between this region and this region? Yes, here's how the path goes. And the colors sort of indicate whether you're going sort of up or down, left to right, front to back, right? And so the color coding, the colors are coded to sort of indicate that. And so these are really lovely images, and this is a different type of structural image which you can get at the same time as getting any of these three images using MRI. Now, a functional neuroimaging is a more sort of maybe a more recent development, and it's I think fMRI is roughly 30 plus years now. So I guess recent is in the eye of the beholder, but there's been a lot of interest in using functional neuroimaging to study sort of cognitive and affective processes. So how do we think? How do we feel? How do we react to different things? These types of things can be studied with functional neuroimaging, and I'll explain how in a moment. So again, there's lots of different modalities here. So PET, it can be used to do structural images, but also functional images. Now, MRI, the same MRI scanner that we just said was so versatile that could be used to create these structural images and these diffusion weighted images, they can also be used to collect functional images. And then we just usually call that functional MRI or fMRI. And that's going to be the primary focus of today's talk. But there's also other techniques like EEG and MEG, and each of these provide a different measure of the brain. And there's sort of pros and cons with each related to sort of spatial resolution, temporal resolution, and sort of the relative invasiveness of the technique. So just to give you a high-level overview of the difference between sort of structure and function. So structural image, I guess, is sort of like a photograph. So if I took a photograph of Yi right now, it would be a description of what she looks like at the moment. But will she look like this tomorrow or even in 5 minutes? Well, she might go dye her hair or, so it could change, right? I don't know. But I could get sort of a high spatial, but with structural images, you can get sort of high spatial resolution, but you don't get any temporal information. I don't know what's going to happen in 5 minutes or in 10 minutes. Presumably, it's going to look relatively similar unless you have some sort of stroke or something. But what this the information this tells us is what the brain looks like at this time. But it's very useful for sort of telling apart tissue type like gray and white matter and CSF and the like. Now, functional images are a little bit different. So with structural images, we can spend a bit of time to try to get a good resolution as possible. So it takes a little time to acquire the data and get it to a good shape. With functional images, we sort of want to rapidly get images. So this is sort of like a movie, right? So we want to get lots of different images so we can look at it in quick succession so that we can see how the brain changes during a short time window. And so because we need to get these images very quickly, they tend to have a lower spatial resolution, but of course they have a temporal resolution and they have a higher temporal resolution. And so the temporal resolution sort of depends on the modality and whatnot. And so one thing that we want to do is we might want to look at relate changes in the signal in relation to some experimental task. Let's say I have a condition A and condition B. During condition A, I tap my fingers. During condition B, I rest. And I want to see, is there a difference between the signal when I'm tapping my fingers and then when I rest? And if so, where in the brain is that taking place, right? And so that's something that we can do with functional images. Now, again, there's these different types of functional neural images. And so at its core, what do we want with functional images? Well, we want to know where activation is happening in the brain. So if I sort of clap my hands, sort of neurons in my motor cortex start firing because I'm moving my hands and things like that. And so um, at its core, I want to get at where are those neurons firing? Well, the bad news is, I could get that if I sort of implanted a sort of an electrode into my brain and got a recording like that, but that's very invasive and it doesn't give you sort of whole brain coverage. But with fMRI and the like, we can get sort of whole brain coverage of what's going on when I clap my hands. But the trade-off is that I can't tell you where the neurons are firing. I can only study sort of byproducts of the neuronal firing. So what happens when the neurons fire is that the blood flow changes. So there's the active neurons sort of feed on oxygen in the blood, and because they're feeding on that, more blood flows to those regions, and it's that blood that we're sort of studying with fMRI. So we're actually seeing a byproduct of the neural activation, which has sort of important implications for things like if you were going to do causal inference, like you mentioned in the introduction. Um, PET, for example, studies brain metabolism, and things like EEG studies electromagnetic signals generated by neuronal activation. And you'll see here with EEG is in that case you're actually wearing a cap where you're measuring signals on your over different channels on your scalp. So these are the locations. So this is a person, that's their ear, and this is their nose, right? And so this is where the channels are placed, and this is what the time series look like. So in this case, you can get a very high temporal resolution, but you'll see the spatial resolution is sort of limited to these channels on the scalp. While with something like fMRI, I can get really like whole brain coverage at a very fine spatial resolution, but the temporal resolution is usually a little bit slower. So those are sort of the trade-offs that we have here. So in general, MEG and EEG tend to be very high temporal resolution, relatively poor spatial resolution. PET has worse temporal resolution, and it's also invasive because you have to sort of ingest radioactive material, and that's how we're doing that. And so in the sweet spot is sort of found here in what we call bold or blood oxygen level dependent fMRI. And so bold fMRI tends to have a good spatial and temporal temporal resolution, and it's also non-invasive. So it's really become the de facto, the go-to technique for a lot of neuroimaging people. So I'm going to sort of focus on that modality, but a lot of the things I'm talking about are going to sort of carry over to other sort of imaging modalities. So if we're going to focus on fMRI, let's sort of get to know the data a little bit better. And so functional MRI is, again, sort of a non-invasive technique used for studying the brain activation. And so during the course of an fMRI experiment, you acquire a series of brain images, so such like the one that I showed before, while the subject performs a set of tasks, or maybe even just rests. That's a very popular, it's a task in a sort to not do anything, right? And so basically what one does is you look for changes in the measured signal between individual images, and you use that to make inferences regarding task-related activation in the brain. And so let's take a look at this a little bit. And so let's say, consider a brain here. So what we do is, let's say that you put the brain inside of a box. You have the brain here, you put it in the box, and then you split that box into around 100,000 different smaller, equally sized boxes. And those cubic volumes are called voxels, or volume elements. So basically what we're doing is we're splitting the brain up into 100,000 sort of equally sized boxes. And each of these boxes, or voxels, contain basically two pieces of information. And so one is, it has a spatial location. It might be in my amygdala, or in the hippocampus, or in your visual cortex. It's located somewhere in your brain. The second piece of information it has is an intensity value. So in this little cartoon, we pull out this voxel in this location, and has an intensity value of 39, for whatever that means, right? And so those are basically the two pieces of information, and that's actually all we really need. And for example, to make an image like this, you don't really think of this, but as a mathematical object maybe, but this is just a pictorial representation of a matrix, right? So this is a slice of the brain, and you can think of this slice of the brain just being split up into a number of voxels. And if the voxel takes a low value, you give it a darker color. And if it takes a higher value, you give it a lighter color. And then suddenly, because you have these intensities and their spatial locations, you can make out these contrasts, and you can make out these areas in the brain, and little islands of gray matter and white matter. So really, at its core, any image like this, any medical image that you see like this, is simply just a matrix. And I think that's an important thing to think about as a statistician, because that gives us an insight into what we can sort of do with these kind of things. So in general, so that's a single image. Well, with fMRI, so during the course of an experiment, we acquire sort of hundreds of these images, maybe once every half second up to 2 seconds, depending on the study. Classically, it was every 2 seconds for a long time, but with sort of modern acquisition techniques, they're becoming increasingly higher temporal resolution. So these days, some of the higher quality data are about a half second or so. And so in this situation, we basically have, again, we have this brain image acquired at time 1, time 2, time 3, all the way up to time t, which time t might be 500 or 1,000 or something like that. And basically, remember here is that this is just a collection, and sort of an array of voxels, and each voxel has a location and an intensity. So if we fix one voxel, the idea here is that that voxel across time should have the same spatial location. If we can find that voxel and fix the spatial location, then all we're doing, if we extract data from that voxel, is studying how the intensity changes as a function of time, right? So basically, we highlight that voxel, we fix the spatial location. And so we look at that same voxel across all the images, and we just look at its intensity. We have a time series all of a sudden, right? And so this time series in this cartoon image, I have this sort of on-off paradigm where, let's say, I'm finger tapping for 20 seconds, resting for 20 seconds, finger tapping for 20 seconds, resting for. So regions of the brain that are sort of active when I'm performing this task, they tend to have higher intensity when I'm finger tapping and lower intensity when I'm resting, right? So it's going up. Regions that are related to this task are changing in tandem with me performing this task. It's a little bit more complicated than that because the change is a little bit slower. There's this thing called the hemodynamic response function, which means it takes a while for changes to be spotted. But at its core, just to get a basic understanding, what we want to do is we want to sort of link tasks to these time series. So at its core, this is kind of a time series problem, which is a comfortable place for a lot of statisticians. But the place where it becomes a little bit uncomfortable is the fact that this is one time series. We have 100,000 different locations. So we actually have 100,000 different time series, right? So this is sort of a time series problem on steroids. And one that's sort of very complicated. So in this type of situation, lots of sort of classic time series analysis sort of focused on a single time series and they sort of analyzed it for a long time. Here you have whatever analysis you do on a single time series has to be sort of scalable in a way that you can apply it sort of 100,000 or more times. So that complicates things. There's also other things that complicate it, which I'll talk about in a second. So in general, the analysis of fMRI data is an example of sort of what we would call a modern big data problem. Omics and some EHR data, are other examples of that. And so basically, the data from each subject consists of tens of millions of measurements, right? So for example, we had maybe 100,000 voxels in the brain. That's 100,000 measurements. That might be measured over 1,000 time points. And you might perform several experiments while you're in the scanner, and so you might repeat the task several times. You might bring in a subject for multiple sessions. For example, if you're doing a longitudinal study, you might bring them in once a month or once a year or something like that. And also the study experiment is usually repeated for multiple subjects. In small labs, it could be on the order of tens or 20 people, and that was the case for a long time. But in modern biobanks, for example, the UK Biobank, they're currently at sort of 60 or 80,000 released participants with a goal of 100,000. And so that's a very, very big study. But you can just imagine the amount of data now that you have, and so the issue with big data isn't really the big data because I guess there's a problem with storage, right? But the complication with analyzing big data isn't the fact that it's a lot of data points. We can sort of deal with that. The complication usually comes from sort of the complex correlation structures that occur. And so in neuroimaging or in functional neuroimaging, we have very complex correlation structures in both space and time. And again, sort of statistics plays a crucial role in understanding the data and obtaining relevant results that can be used and interpreted by neuroscientists. And so I wanted to spend a little time sort of exploring these correlation structures. And so here's one thing that kind of shows some of the complications. So this is an example of one slice of volumetric data. So I'm just taking one slice of this three-dimensional cube that I have. So that would be, I'm fixing the z coordinate and I'm just looking at the two-dimensional grid of points here. And so again, I told you it would look something like this, right? And so maybe lighter values are higher intensity and darker values are lower intensity or whatnot. And so this is just basically looking at a single time point. But the interesting thing, if you look at the brain, is that it's very folded, right? It's like a tissue that's crumpled up, and you see this intrinsic folding here in this sort of pinkish things. And if we highlight that, you sort of see that if this voxel is next to this voxel here, the direct connection between them is very far away. So, for example, for me to go from this voxel to this voxel, I have to go all the way around this folding to come here, right? I can't jump over this other tissue type to do it. So this complicates things about like evaluating the spatial correlation, because the spatial correlation between two adjacent points in this direction might be very strong, but in this direction they might be pretty weak because they're actually pretty far away from each other in sort of actual time. If you were walking, if you were flying, they're very close, but if you're walking, you have to walk all the way around like this, right? And so the inherent structure of the brain sort of complicates the spatial resolution. So one solution to that a lot of sort of modern techniques are using are sort of what's called inflating the brain. So what we do is you sort of uncrumple this, and so you just blow up the brain into inflate it until you can eventually into like a surface like this. And once you're on the surface, then these 2 points are going to be very far away from each other while they're close together in the volumetric space. So the surface is a more accurate sort of depiction of things. So sometimes when people do spatial modeling, they like working on sort of on surface data instead. In that case, the grid-like volume data, often become something like a mesh like this, which is sort of not, which is sort of irregularly spaced depending on sort of the information content. So this is sort of another way of looking at the data. So I present it as volumetric data, but you don't have to look at it that way with these sort of these boxes. You can look at it as these meshes based on this. And so for some type of statistical problems, this might be a beneficial way of looking at it. And for others, this might be. And so you can sort of move between those different areas. Uh, so there's a lot of spatial correlation in the data. So this is just a plot of activation across the brain. And you're seeing that there's a lot of sort of similar activation in similar brain regions. And you're seeing that it's not like a salt and pepper pattern. It's a very sort of smooth transition from yellow to red and whatnot. And so this is sort of implying that there's a lot of spatial correlation and nearby vertices or voxels are behaving in a similar manner. There's also a temporal autocorrelation. So if I look at some of the data and I fit sort of an autoregressive model to it and try to find the optimal P in an ARP process, the optimal order of the AR process, you'll see that it varies across the brain as well, but it's non-zero pretty much everywhere. And so both of these things sort of conspire. So there's a kind of a lot of spatial correlations. There's a lot of temporal correlations, and sort of understanding both of those is an important thing. So that's hopefully gives you sort of a highlight of a little bit of the data structure. The next thing I wanted to focus on are what are some of the sort of research questions that we want to address. One of the sort of earlier ones is something which I call brain mapping, is that can we identify areas of the brain that activate in response to a certain task? So if I tap my fingers and then rest, tap my fingers, then rest, can I find areas of the brain that are active in response to that task? And so basically what I try to do then is in a way, it's a little bit, again, a little bit more complicated, but I want to sort of correlate the signal extracted from each voxel with the task to see where I can find that sort of relationship. And the end goal is to get these sort of brain maps, which are showing areas of the brain that are active. And so these brain maps are basically showing you p-values of statistical tests. So in every voxel of the brain, you look at the relationship between the task in question and the signal that you can extract, and then you perform a statistical test. And any statistical tests that are above a certain threshold, you deem active, and then you color code them, orange, and everything below you just leave sort of blank here. And so this is a way of sort of presenting things, and again, this is kind of more complicated than I'm presenting it, because again, you're performing basically 100,000 different hypothesis tests to get these maps, and so that becomes a huge sort of multiple comparisons correction problem and whatnot. So there's a lot of things, there's a lot of modeling issues and sort of multiple comparison type issues that come into play when making these types of maps. But these types of maps were for a long time sort of the bread and butter of functional neuroimaging. And people got very excited about this, and they say, well, I can do different psychological tasks and see what areas of the brain sort of light up, as they call it, and and so, you could say, well, what areas of the brain are active when you're sort of solving a math problem? So you could have people solve math problems in their brain, and you could do these statistical tests, and you can sort of figure out where it is. This map here is what's called the working memory task. This is a 3 back minus 1 back. So basically, you're basically seeing these images in rapid succession. And if you see this, the 3 back is that if you see the same image that you saw 3 images ago, you're supposed to press a button. So it gets kind of hard after a while. And this is basically looking at sort of activations that are related to that. Um, More recently, in the last sort of 15 or so years, people have gotten increasingly interested in connectivity analysis. And this is the question of how do different brain, so we know that there are certain brain regions that are active when I perform a task, but the question then becomes, well, how did these different brain regions interact with each other? And how does this depend on sort of different experimental conditions or sort of behavioral measures? And so in this context, we usually discriminate between what's called functional connectivity, which is sort of defined as the undirected association between two or more fMRI time series. This could be the correlation between, or the partial correlation between two different regions of the brain. And that can give you a rise to a graph like this, that maybe this is some graph from the partial correlation between a number of different brain regions. The other type of connectivity we often talk about is effective connectivity. And so effective connectivity is the directed. So here we might write sort of the equivalent of a DAG where we're going activation here leads to activation there, which leads to activation there. And this is kind of one of the holy grails in connectivity because you want to sort of understand these things. And this is where sort of causal inference and the like come into play. This is very complicated because of, as I mentioned before, this is sort of an fMRI and the like are indirect measures of neural activation. So there's a little bit of a lot of assumptions that need to be made to make these sorts of models, but they can be made, and this is an area of research. And so a lot of this research has been driven by something called resting state connectivity, and that's looking at the brain while people perform no task at all. Basically, what they're doing is they're looking at a crosshair usually, and just basically letting their mind wander. And so this has proven to be a very fruitful area of research because even though all our brains are different, when our brain wanders, it tends to incorporate similar regions of the brain. And so this is a seminal paper from Michael Fox et al., where he took a seed region. He took a time series from the posterior cingulate cortex, the PCC here, and basically he correlated it with every other voxel in the brain. And what he saw that it was highly correlated with this medial prefrontal cortex here. And so in very distributed regions of the brain, it was highly correlated. And this was very sort of reproducible in subjects. And this has now become known as sort of the default mode network. And so usually when you're doing nothing and your brain is just wandering, your brain is in this default mode network and it's using these different brain regions are communicating with each other in a very synchronized manner. So if you look at this is the PCC in yellow and the MPFC is in orange, that they're moving up and down in a very sort of synchronized manner. Interestingly, if you look at this interparietal sulcus here, it's actually negatively correlated with the PCC. And this is kind of a part of the dorsal attention network. And so this is usually active when you're focusing on tasks, so sort of the opposite of mind wandering. So this sort of shows some of the interest in the default mode network. So when you're mind wandering, the default mode network is really active and it's sort of anti-correlated or negatively correlated with these attention networks. And then once you spring into action and start doing something, these attention networks become really active and the default mode network is suppressed. And so they're sort of going up and down in an interesting way here. And so in general, these are some of the most commonly identified resting state networks, and the default mode network is one of those. So it's a pretty interesting area of research. More recently, and this is a lot of statisticians have worked on as well, is people have become interested in something called dynamic connectivity. Because if you imagine being in a scanner for 10 minutes and just say, let your mind wander, you can sort of imagine if you just sit in your chair or lie in bed for 10 minutes and think, your mind is going all over the place, right? In the beginning, you might be thinking, oh, this is an interesting experiment. I'm in the scanner. And then it's like, oh, on the way home, I have to go pick up some bread and I have to pick up my kids or I have this paper revision I have to do. And so you're sort of jumping between different thoughts and different sort of states in this thing. And so there's been some interest in sort of trying to figure out whether or not things like this functional connectivity change over time. And this is sometimes called dynamic or time varying connectivity. And so here's a paper from a student of mine, Yuting Xu, trying to find change points where sort of the connectivity structure between different brain regions change. And so there's a lot of techniques for using that, like hidden Markov models and the like. Here's another version that uses sort of clustering, which basically shows that this is from Vince Calhoun's group, who do a lot of work in this area, that basically showing that the brain is toggling between these 5 different states at every time point. And so they start out in state 3, then they move to state 2, then to 4, then to 5, and then back and forth. And so it's been really interesting to sort of see whether or not people with certain sort of disorders tend to be in certain states more often than others, or if certain people with certain disorders tend to switch states more often. So for example, in some work we've looked at kids with ADHD tend to switch more often, which is than typically developing kids, and that might be reflective of sort of attention issues and the like. Um, Finally, if I do a connectivity analysis and make a graph, it might look something like this. And if you're asked to sort of interpret this, that's awfully difficult because it's just a muddle of different arrows between them, and it's very hard to. And so sometimes it's interesting to sort of get summary statistics out of these graphs. And to do that, a lot of people have gone back to sort of graph theory, which has a long history in mathematics, going back to Euler and things like that, to investigate sort of organizational principles of the brain. And so it turns out that there's sort of clear topological organization in the brain. So the brain sort of behaves like a small world network, much like social networks and things like that, in the sense that they tend to communicate a lot with nearby regions, but also every now and then with long range, which helps everyone be sort of connected to each other. And so often these connections are dealt with by highly connected network hubs. And so why is it important to know where these hubs are? Well, if you get sort of a stroke or a brain lesion and you knock out one of these hubs, you're probably going to lose, if you survive, you're probably going to lose a lot of sort of abilities, right? Maybe the ability to talk or to move or whatnot. But if you knock out a region that's not connected to important regions, you might not even notice that you have a stroke. So this is very important for sort of understanding these types of things and sort of the resiliency to damage and stuff like this. And also comparisons of network topologies between groups of subject can reveal sort of connectivity abnormalities related to sort of psychiatric disorders. So here's just a kind of a cartoon image. We have this little network here. We can make an adjacency matrix showing which regions are sort of connected with each other. And we can basically calculate sort of things like the path length. How long does it take to get from B to D? And, clustering coefficient, how related are, so C is connected to every one of its neighbors here, right? But B is only connected to A, and so it's about, so if B is knocked out, it's not as serious that if C is knocked out or something like that. And so those are the kind of things that we can sort of investigate. The final big area, sort of brain mapping, connectivity at all, is prediction and classification. And so this is a kind of an increasingly important thing. So can we use brain activation to predict whether a person has a disease, yes or no, how old they are, whether they're in pain or not, or whatever. And so in this case, we take all the brain activation and we fit sort of a machine learning or AI model, and we try to predict some sort of outcome. And so basically we want to find some function of the brain that's predictive of the outcome. And so here's one example that we've done is which is called the neurologic pain signature, which can predict physical pain using brain activation. And so basically, these are just the weights from a linear it's a type of support vector regression. And so if you have a lot of activation in these yellow regions, these tend to be weighted highly, and you tend to report more pain. If you have a lot of activation in these bluish regions, it tends to suppress the pain report. And so, you know, being able to use this and sort of transition it to a clinic is sort of an important step that we're sort of working on. Finally, you know, I've talked about sort of imaging and things like that, but one of the more hotter areas is sort of taking sort of interdisciplinary approaches and combining different types of modalities with each other. It can be different imaging modalities. For example, you know, I said that EEG had high temporal resolution but low spatial, and fMRI the opposite. What if I combine them? I could have my cake and eat it too. And so people often want to do things like that. Also, neuroimaging data is increasingly being combined with sort of non-imaging modalities such as behavioral data, but also with genetic data. So something called imaging genetics, which is sort of trying to link sort of genomic finding with differences in brain. So are there people with different sort of SNPs? Is that related to different activations in the brain? And so this promises to be a really important topic of future research, but to be sort of fully realized, you know, novel analytic techniques will be needed because this, if imaging is a big data problem and omics is a big data problem, this is sort of a big data problem squared. So it's a really hard to work on. But in general, this is something that we often want to do is basically take brain function and link it with behavior and genetics and try to figure out how this all connects with each other. So a lot of studies are now working on these different connections, and this is sort of a fertile area for statisticians, but again, it's a very complicated one, and so in sort of initial studies, people do shortcuts. They might take a couple of derived phenotypes from the brain and then look at all the SNPs, or a couple, they might take a polygenic risk score from the omics and apply it to the brain and things like that. But since this is a kind of an AI focused thing, I also wanted to sort of talk a little bit about how AI has been used in neuroimaging. And of course, like everywhere else, AI is very hot and people have been trying to apply it to a variety of different techniques in areas in neuroimaging. And so I basically made 4 different areas where I think that they're pretty widely used. One is image processing. So this is segmentation, reconstruction, outlier detection, denoising. This is basically the pre-processing of the brain. Another is image generation. Can I basically make realistic fake brains that I can sort of share more easily than regular brains because of PHI concerns and privacy concerns, or that I could use in simulation studies. Prediction, can I basically predict sort of how much pain a person is feeling and do a deep learning model to do that? Modeling of sort of biological networks. So these neural networks are sort of based on what was assumed to be the brain, right? And so can comparisons between artificial and human networks provide insights into neural systems? And so I'm just going to talk briefly about the first and the third point. So in the first point, that's probably where AI has had the most use. So AI is very good at sort of segmenting the brain, and that can be important for sort of just segmenting between gray and white matter or finding where in the brain there's a lesion and things like that. It has also been good at sort of providing alignment of scans to sort of templates. So oftentimes everyone's brain is a different size. So we often need to put it on a sort of standard population template to compare across subjects. AI is pretty good at doing that and enhancing low quality scans using generative data. I think a common theme of all this is that AI is really good at learning algorithms because there's algorithms that are used for all these things, and it's easy to create training data. So I can just sort of segment thousands of brains and put it into an AI model, and basically the AI can learn the algorithm that does it and do it faster and maybe even more efficient in some ways. So AI has had a lot of success in that application in imaging. And one of the reasons is that it's very easy to create a lot of training data and the like. an area where it has been less successful is sort of predicting cognitive states and clinical outcomes and saying who has Alzheimer, who doesn't, and the like. And in general, these AI or deep learning models don't outperform classical machine learning for behavioral prediction. And they're generally haven't been shown to be worthwhile because there's a trade-off in interpretability. So here's one paper that shows that sort of deep neural networks and sort of kernel regression are basically comparable, while the kernel regression is much more interpretable. And so for neuroscientists, interpretation is very important because remember when I was looking at that map of the pain signature, I can interpret that and make neuroscientific statements based on it. But that's sometimes harder with these more complicated AI models. Um, one successful application of deep learning has been in predicting age, and that's been kind of actually a hot topic recently, and that's sometimes been called brain age. So basically, I can look at your brain and predict your brain age, and what people are sort of interested in is what's the difference between your actual age and your brain age, and that's sometimes called the brain age gap. And it turns out that several disorders have been linked with sort of prematurely aged brains. And in this case, a lot of the best sort of algorithms for predicting this are based on a combination of AI and sort of large scale neuroimaging data sets that sort of learn patterns of age-related changes in the brain. So that's a kind of an interesting area that a lot of people have sort of worked on and found sort of utilities with. So I think that just in general, some of the challenges with applying AI to neuroimaging data in general is, again, the lack of interpretability due to model complexity. I mean, that's getting better in AI, but it still has a way, it's still much easier to interpret linear models, right, than these. There's also AI is a little unscrupulous, right? There's prone to learning shortcuts or confounding information that may not be relevant to the underlying task. And I'll give you one example here is that um, there was a competition to predict that there was 100 kids with ADHD and 100 typically developing kids. And so there was a competition to sort of predict who had ADHD and who didn't, building a sort of a machine learning type model. And it turned out that the group that won basically created a glorified motion detector because kids with ADHD tend to be squirmy and move around more, right? And you don't need $1 million scanner to do that. You just need to sit with the kid for 5 minutes and sort of if they're under a table after 5 minutes, they probably have ADHD, right? I mean, it's that so AI doesn't know that it's supposed to focus on sort of neurological or interesting things. It takes whatever information it can, right? And because it doesn't know any different, right? And so uh, that's what I sort of mean by confounding information, taking that into, and so that's a risk with these AI models. Of course, they don't always generalize to new settings. This is becoming a bigger deal. So research limitations, training state-of-the-art foundation models is becoming expensive and inaccessible to many groups. I think a lot of computing needs to be done on the cloud. So some of these UK Biobank won't be accessible on individual clusters anymore. You're going to have to do it through cloud services, and there's costs associated with that, which sort of complicates things. And of course, there's biases based on who we sample, and the machine learning algorithms learn those biases. I think moving forward, there's a few areas where I think AI can play an important role. One is in data synthesis. It's basically performing a meta-analysis. Can it search the web for all studies of working memory and find a consensus sort of brain map of that? Can it create different analysis pipelines that take into consider different things? Can I drag in an image into a browser and it gives me information about, oh, this person has an enlarged amygdala or has a tumor there. That seems to be something that AI can probably do very well in a couple of years, and it might be something moving forward. I think the fusion of population level data with smaller data sets in a principled manner using transfer learning, I might be able to train models on these large UK biobank and then apply it to my study of 20 participants because those 20 participants, their brains have a lot of similarities with those 100,000, but then they also have idiosyncratic things. So we want to sort of utilize the similarities and then let the idiosyncratic stuff come out. And so there's probably going to be nice ways to use AI to do that. Again, I mentioned generating synthetic information, which allows sharing of sensitive data, and also just acquiring better images with shorter acquisition times. And these are things that AI can be potentially used for. Now, after hearing this talk, you said, "Well, I want to try to do some of these things." How do you get started? Well, I think that's an important part of this talk as well. So I think if you want to get started in functional neuroimaging or any neuroimaging, basically any data problem, uh, the first step is basically to get the data and just get your hands dirty and figure out what the data looks like, its complications and things like that. There's really no shortcut for that, I think. And the nice thing about the neuroimaging community is very generous. It shares data and it shares code and it shares software and things like that. So there's multiple initiatives to share medical imaging data between researchers. And these initiatives acquire data from different sites, scanners, imaging protocols, and making them a rich resource. Here are some examples. So the Human Connectome Project, it was originally on about 1,200 people, but now it's like through the whole lifespan and lots of different psychiatric conditions, and it's on several thousand people. The UK Biobank, I already mentioned, is going to be 100,000. This is autism brain imaging. This is ADNI for Alzheimer's. This is the ABCD data set, which is 10,000 kids between, as a longitudinal study of 10,000 kids between 10 and 20. So they're studying these kids in the wild and scanning them every year as they go through puberty. And a lot of those kids are going to sort of experiment with drugs. Some of them are going to develop sort of psychiatric diseases. Some of them have onset after puberty and things like that. And so this is going to be a great way of studying that in the wild. And A2CPS is one that I'm involved with. We're studying 3,000 people that have an acute pain episode. They have a surgery, so we scan them before the surgery and then 3 months after the surgery. And about 25% of people who have the surgery are going to develop what's called chronic pain. And so we want to look at sort of brain measures as well as omics measures to see, what makes somebody resilient to chronic pain versus prone to chronic pain. Um, so it's easier to get that data from the web. You might have to sign a data use agreement, but how do you read in the data? Well, the good news is there's lots of free open source software packages. The big three has traditionally been statistical parametric mapping, or SPM, AFNI, or FSL. FreeSurfer allows you to do the surface-based analysis. That was when I said that you blew it up onto like a sphere and do that. More recently, there's been a lot of work in Python, and so NiPy is trying to sort of integrate all these things into a single framework that you can call via Python. So if I were starting today, I would probably start in Python because you could use something like NiPy and access all these things through a single interface. There's also an R-based platform made for statisticians, which is called Neuroconductor, and the name is a play on Bioconductor, which is sort of a similar effort for in the omics world. Um, so in general, just to finish up here, the field surrounding functional neuroimaging is sort of constantly evolving. And, again, more and more ambitious, larger studies are being performed each day. New research questions are opening up, and so there's a great demand for quantitative people. And so you should just get started if you're interested in it, because it's really sort of a gift that keeps giving. There's lots of interesting problems, and they get more and more complicated and more interesting. So looking forward, I think some of the most important questions will involve things like translational neuroscience. How can we move some of these techniques from research to the clinic? And that's an important thing. Again, transitioning to these large, diverse lifespan data sets. How can we utilize these biobanks to help inform what's going on in our smaller studies? Sort of merging different imaging and non-imaging modalities. This is imaging genetics. Integration with neuromodulation techniques. There's things like TMS and TDCS that can sort of, you basically give a little electronic current, and that allows you to sort of suppress or activate different brain regions. If you want to do sort of causal inference on the brain, that's a great way of sort of doing that. but there's some, it's still in process trying to make these techniques work with high temporal spatial resolution, and of course incorporating AI. And so again, statistics plays an important role, but I think I always sort of say that, you can make the biggest impact if you sort of really embrace the interdisciplinary nature of the problem and sort of move outside of your comfort zone and sort of get to know the data and the problems that the neuroscientists are interested in. So I really think that makes the world of difference. So I just want to start out with thanking you and then mention that as if you think this is interesting, again, we have this Coursera class. It's also available on YouTube. So if you go to YouTube and write Principles of fMRI, you can get the whole class. It's a bunch of different modules, and and there's also sort of a book coming out next year based on this. So I think that's what I have. I don't know if there's any questions? Um,
Thank you, Martin. I don't see any question in the chat box yet, but if you have any questions, so please feel free to raise your hand and then I will call your name and you can ask question. Um, so maybe may I ask a question? I think
Absolutely. Yeah.
so when I think to get started, so I think the first step is to download the data and then get your hands dirty. But when I first started like working on neuroimaging the field, and then I think the hardest part is to do the data pre-processing. So like pre-process the data because there are so many packages available, and then there's really no gold standard in terms of gold standard in the field in terms of doing data pre-processing. I'm just wondering if you have any suggestions and then where we should get started, or is there a like a standard data set we can start?
Yeah, I mean, this is a good idea. I mean, I think as a statistician, I probably would recommend to download already pre-processed data to probably start with. So you can go to places like Open Neuro and even go to like the UK Biobank or the Human Connectome Project. They have mostly pre-processed data. That's probably where I would start and get comfortable with sort of reading in the data and looking at sort of correlation structures and things like that. But I do think at the end of the day, um, there's a lot of your data from coming off the scanner to being in that pre-processed step has gone through a lot of different hoops and things like that. And so I think it's important to sort of understand that. So as you sort of mature as a researcher, I think it's useful to sort of use these techniques. And there's things like fMRI prep is something that's from in this Python NiPipe kind of universe. Uh, and and that allows you to sort of create pipelines, and you can sort of play around with that and see what the effects are. And so that would sort of be a kind of a later step.
Okay. I think I saw a
Oh,
hand.
there's a few here in the chat now. What does it mean that the fMRI bold oxygenation dependent? Oh yeah, so bold is blood oxygenation level dependent. So basically, it's just a type of you can it's a type of contrast that we're studying in fMRI. It's basically just um, when the blood has oxygen, the blood contains oxygen and when the neurons fire, the oxygen is consumed by the locally, and there's an inflow of oxygenated blood to that region to sort of replenish. And that's what you sort of track with bold fMRI. So it's a particular type of fMRI that tracks how the blood is flowing in your brain in reaction to neuronal activation. How has the literature on functional brain connectivity models dealt with extreme dynamicity of this problem? Also, is this a potential reason why AI is failing in prediction because the dynamic nature of connections make it extremely data driven? Yeah, I mean, I think AI is not only, I think the AI is failing in prediction for two reasons. One is that the sample sizes are still relatively low. People haven't been very good at sort of augmenting, doing data augmentations and getting bigger sample sizes. I also think a lot of the things that people tend to predict are very hard to predict. For example, things like autism is on a spectrum, right? And even two doctors can't agree on a diagnosis. The AI isn't going to be able to figure out if it has crummy sort of training values. So some of those things are very, very hard to do. Um, but in general, how is the functional brain connectivity dealt with dynamic nature? Well, there's a lot of things. Again, I've mentioned these change point models. There's also sort of hidden Markov models have been used a fair amount, and sort of the akin. I think that maybe AI could be used in this place, but you'd have to figure out a way to sort of train it in a good way.
Elena, so you have a question?
Yes, thanks. Well, first of all, fantastic talk. Thank you so much. Going back to the part where we talk about more like graph theoretical analysis of the networks in the brain, I was wondering if there's like conventionally a set of like maybe anatomical, like if there's an anatomical meaning to what those nodes represent.
Yeah,
Basically, what is the like what are those nodes and how is the variation in the literature in the
yeah,
real?
that's a great question. I sort of brushed that aside. And so often, again, the brain consists of maybe like 100,000 or so voxels, right? But when we do graph analysis, we often do use what's called a parcellation, which is basically maybe I break the brain down into 200 regions. And one region might be the visual cortex, and that consists of thousands of voxels. And then another region might be the amygdala. And so you basically sort of cut the brain up into sort of 200 regions. And usually it's between those regions that we do sort of graph analysis between. And so each of those regions have sort of inherent meaning in the sense that the visual cortex processes visual information. There also, some of these regions can be grouped together into networks. Like I was talking about this default mode network. The default mode network consists of multiple regions that are sort of distributed across the brain. So usually we do this sort of thing that the voxels can be combined into regions, and then the regions can be combined into networks. And so what we're often doing is we're making these graph structures on the networks, but on the regions or the networks, and then sort of being able to say, well, hey, look, the default mode network is really highly the regions in the default mode network are really highly connected with the regions in the frontoparietal network or something like that. And that allows for sort of an interpretation and that's sort of necessary. Yeah. So, and so what parcellation should you use? Well, there's lots of different ones and people have their favorite and things like that.
Thank you so much.
So Dan, also you commented about the question as well a little bit.
Yeah, all I was saying is there are structures in your brain, like the caudate, putamen, nucleus accumbens, all thalamus, just like there are structures inside your torso. You have a heart, and you have a liver and a kidney. They're definitely defined structures, is all I was saying.
It's already past one. I'm not sure if Marty, you can take another question
Oh
because
yeah, that's fine to take another question then. Yeah.
Kaiser Fang.
Hi, I guess I'm taking your previous answer in the other direction, which is that how dependent is the number of voxels and kind of the granularity of these voxels? How would it affect the analysis? Because it seems like once we have settled on these voxels, and everybody has a presumably different size and shape of the brain, that we have somehow assumed that the same voxel is the same voxel everywhere.
Yes. So again, so this is part of this pre-processing that Yi does because all our brains are really different because the size of our bodies are different. Similarly, our brain sizes are really different. So what people usually do is they actually warp every person's brain onto a sort of an anatomical brain. And so this is called normalization. And so basically, at the end of the day, every brain should overlap with everyone else's. And so if I pick a voxel in my brain that at a certain coordinate, the hope is that that would be the same voxel in your brain if the registration has worked. Now that it doesn't always work that way, and there's sort of some residual differences. So that's one of the things that, for example, that's one of the research areas that I work a lot with, is sort of functional differences in the brain. It might be that your amygdala looks like this, but mine looks like this, even though we've registered that. And that's something that we're quite interested in, is does the size of a certain brain region or how much of it, or which parts of it that are utilized, sort of inform sort of behavior or outcomes and things like that.
Thank you.
Great. I think that will wrap up our seminar. And then finally, thank you, Marty. And then finally, just give a preview. So our next seminar will be on May 9th and then between 12 to 1. So we will have Dr. Ingo from Emory talking about fMRI again. So we already lined up a series of seminars to introduce different type of imaging modality, like Martin introduced today, like EEG. So we will have an EEG introduction in June, and then later DTI, and then structural imaging, and then PET. So yeah, stay tuned, and then thank you so much for joining us, and I hope to see you next time. Yeah, bye.
Great.
Thank you, Marty.
Thank you.
Bye, thank you.
Thank you very much. Great
Good
talk.
talk,
Thank you very
Martin.
much. Thanks, everybody.